All right. It's getting late in the day, but I want to get all this out so that you can watch it anytime between now and Friday. And also, I'm going to be talking about food a lot because our subject is the proteins that you find in food, fibrous proteins that hold an animal together. That said, I have an incentive not to ramble on too much. You know, the thing about wanting to tell you so much about this subject is that I go on longer when I don't have the time constraints of class. So I'm sorry, I'm trying to work on that, but I'm also keeping in mind that we've had 50 minutes of lecture recorded already for lecture 9 to be watched on Wednesday. So I'm going to do about another 30 minutes here if I don't faint from my low blood sugar first. So when we're talking about protein structures, once you find the structure, you actually should talk about, you can use X-ray crystallography to figure out what is the tertiary structure. We've talked about what's the primary and secondary. Now we can do tertiary, and if you have a multi-chain assembly, you can talk about quaternary structures. By the way, sometimes I misspell it as quaternary. It's really quaternary. Sorry, I definitely won't mark it wrong if you write it either way on your test. So globular proteins are what we work most with because they work well as chemicals. They work in the cytosol, you know, they fold up, they work in the blood. They're easy to work with in a test tube. Most proteins that we've worked with in the lab are globular. If you take a single chain of 585 residues, that's how big human serum albumin is. If it was all beta conformation and nothing else, it would be one long string. You can take the angstroms per residue for beta and multiply it out for yourself. If you took the primary sequence of 585 residues, stretched it out to beta conformation, it would be 2,000 angstroms long and 5 angstroms wide. That would be good if you need a rope, but it's not good if you need an enzyme. An enzyme has a defined structure, floats around in the cell, moves from place to place. So, beta conformation is clearly not what human serum albumin does. Well, okay, it's not beta, maybe it's an alpha helix. If you take an alpha helix, it's curled up a little more, but if it's only alpha helix, then that ends up with something that's 11 angstroms wide, that's how wide the alpha helix is, and 900 angstroms long. That still doesn't seem like something that can float around in solution without getting tangled up. So most proteins that do the functions that we're interested in, that do chemistry, are actually going to be folded up versions of beta and alpha. And you know you've got to have some turns and loops in there that take the beta conformation and break it and fold it back in on itself. Same thing for alpha helix. This right here just shows you most proteins have to be more globular. This one is almost as wide as it is long. It's 30 angstroms wide and 130 angstroms long. That's like a sphere. And that's what most proteins are. The question is, how are the beta and alpha secondary structure elements arranged within that sphere? That's what X-ray crystallography, cryo-EM, can tell us. So there are some proteins that do need to work in a fibrous way. Proteins that tie the body together, for example. Those proteins are going to be like the beta conformation or alpha helix example. If you have a 585 residue collagen protein, it's going to be very long and thin because collagen is designed to be a rope or a gel, tying things together. So, we're going to talk about fibrous proteins. I'm going to talk about 2 of them in this lecture, and we're going to pick up with the 3rd on Friday. So this is a fibrous protein. That's the hint I'm going to give you. I've already said the name of one of them. Which protein do you think it is? But it's not collagen. This is a biological microscopy structure. So if you think about it, what grows out of the skin and looks like that? These are actually eyelashes. So this is hair. And the hair protein is keratin. Definitely a fibrous protein, made to be a long wavy thing that grows out from the body for whatever purpose, for heat regulation or whatever. What we're concerned with is, okay, what is a strand of hair? It's a lot of protein. How is that protein arranged? Here's a close-up view. You can see it has some scaly parts. And below this level, we can find out that there's thousands and thousands of protein strands that are braided together. So when you're braiding hair, braiding hair is making a coiled coil of a coiled coil. You're actually got several layers of coils. It's kind of coils all the way down there. So that's the interesting thing. Fibrous proteins are used for structural functions, and so a lot of scientists are actually looking at how they work because they have been really optimized by evolution. They work really well. So maybe we can understand about how materials work to hold things together. That's why when you have something like this, which is people looking for biological composites, how can I make a material that is like nature, but I've made it with chemistry, and it can have some cool properties like nature materials do. So these scientists who work with biological composites can work with mineral or sugar or protein, and the three proteins they work with are collagen, keratin, and silk. So those are the three proteins we're going to talk about. So what's inside a hair? Like I said, there's a lot of strands of protein, and if you look down, if you sequence it, you get a sequence, and it's alpha keratin. If you find the secondary structure, it's get a lot of alpha helices and nothing else. It's really two alpha helices twisted around each other in a two-chain coiled coil. Those are arranged in a protofilament, which is arranged in the protofibril, which is arranged in an intermediate filament, and there are some cells thrown in around it. Guess what? All these terms are biological terms. What I care about that you know is alpha helix and two-chained coiled coil. Just realize that alpha helix itself is a coil, and so a coiled coil is a coil of the two alpha helices. So it's a general term. You can have a coiled coil of three alpha helices and another thing, or it doesn't even have to be an alpha helix. It could be another kind of helix, another kind of coil. It's a general term for a twist around another twist. Here's a biochemistry journal cover that shows three coils, and you see how each rope is itself a coil, and this is a model for another a fibrous protein, but you can see how each one is made of three strands, and you can arrange them different ways. Alright, so that is keratin. What is this, though? This is a microscopy. Pull on your biological knowledge to think of what is this. This is something that is another fibrous protein. I've already mentioned it, so you might be able... actually, I think I'm leading myself wrong. This is actually keratin as well. But this is keratin arranged in a feather. So I jumped to collagen too quick. Like I said, I need lunch. But this goose feather right here is what this is. This is also keratin. You can arrange keratin into a rhino horn. Your fingernails are keratin. So keratin can be a material that's not just a long strand like hair. It can be toughened. It can be arranged in a tough way for something like a toucan beak. The toucan beak has been optimized by evolution to be very light so the bird can fly, but very strong so the bird can eat. So if you look at it, you've got these keratin layers. What is it about the protein? That's protein. How is the protein arranged to be so strong and yet so light? Seems like something like this would be cool for airplane technology or other uses. Well, one of the ways keratin is so strong is it's linked together with covalent bonds. One of the ways that we can find out is that we can actually break it with reducing agents, like, again, DTT and BME. I'm mentioning these again because this is the kind of important information. What chemicals can you use to change a protein structure? Well, you can use them to change hair structure, because if you add them to a strand of hair, it breaks all the disulfides, then you curl that strand of hair and you take them away. You oxidize the bonds. The hair remains curled. Now, can anybody tell me, if experience with hair salons, there's a certain smell sometimes when somebody's getting a perm. That smell is the smell of a reducing agent, because that's what you're doing. You're reducing their hair so that you can then curl it and oxidize it in a new configuration. And you're doing that, you have a molecular level thing that you're doing. So that's pretty cool. Hair removers work by being really strong reducing agents that will basically do two things. So this is a hair remover. I think this is the basis of Nair, but it's a related product if it's not Nair itself. It's calcium thioglycolate. So what it really does is, you see the SH right there. it acts as a reducing agent. It breaks the disulfides. But it also swells the hair, loosening the fibers. And it also does one more thing. Because it's a base, it swells the hair. And it also deprotonates the thiols. So once the thiols form, they become deprotonated, and it makes it harder for it to reform, makes the whole hair strand fragile, and the hair is easily broken apart in that case. So it's important that you have the proton around for the hair remover to work. What's really one of the things about Nair is that Nair will break hair, but it won't break cotton. Cotton is also a fiber. It's actually not a fibrous protein, but it's a fibrous carbohydrate. It's not dependent on disulfide bonds. Therefore, a reducing agent will not affect it very much. So here's hair, and hair chemistry means it's a protein. If you have something that binds a protein, it will also bind hair. If it colors protein, it will color hair. And what's interesting about these molecules, these are color molecules that can be used as dyes, so they'll get into the hair, they'll stick to the hair, and they will recolor the hair. What's cool about this dye, and it actually comes from flowering plants, anthocyanins, and it's like hydrangeas, by the way, have this. Hydrangeas are red in acidic soil and blue in basic soil, more basic soil. And that same thing will work with this hair dye. They developed a hair dye based on this, which is pH 4. It has this structure. You take away the proton and it turns blue. It's red when it has the proton, it's blue when it doesn't. So you dye the hair at pH 4, you get this nice red color. You dye it at pH 6, you get this nice blue color. The thing about this is where they get the dye from. Like I said, this comes up in plants. So they're making the hair dye not from some industrial process, but they're taking blackcurrants, which is used to make a liquor, which apparently is a big deal in the UK. They like it over there. And so, but they have all this blackcurrant mash left over. So this is an idea to turn that mash into a red or blue hair dye, and this will stick to hair because it's a protein. Okay, so that was this. What is this? It looks like hair, but you might guess, because I like being kind of tricky sometimes, it is not. This is another fibrous protein. This is collagen. And this is collagen where you expect to see it. A ruptured tendon is made of collagen. You probably have heard that from biology classes. Collagen is a tough rope that links things together, as opposed to the, hair is not as strong as collagen. So hair is not as strong as tendons. Therefore, keratin is not as strong as collagen. Why is collagen stronger? Collagen, like keratin, can be arranged different ways. It can be arranged in sort of flakes, and it can be a major component of skin. So this might look like a picture of a croissant, but it's actually a picture of skin and the layers of skin that come off and they become dust. Okay, but this shows you the layers of flaky collagen that's inside skin. So if you zoom in on collagen, you find collagen in tendons, skin, and also in bone. It has slightly different arrangements in these three biological locations, but underneath it all, it's the same fundamental protein sequence that works the same way. When you get down to the microstructure, you can see that it's these fibers that are like 10 to 100 nanometers wide. And if you look down in the fibers, they're made of all these linked little fibers. When you get down to the little fibers, finally on the nanometer scale, 1 to 10 nanometers, you start to see that they are helices. But you should look at that and you should see that doesn't look like an alpha helix to me. It looks too stretched out. You'd be right. So you've got the molecular structure of this different kind of helix. When you get down to the 1 angstrom level, you're seeing individual atoms, and you see that it's actually got a pretty simple primary structure. It goes glycine, proline, hip. What is hip? It is a 21st amino acid, and it is hydroxyproline. But the basic structure of collagen is that it's got glycine, proline, hydroxyproline over and over again to make these super long chains that end up being super tough because of the way they weave together. So this is the way your book puts it. you can see a single strand. You can see a single strand with all the atoms shown as spheres, and 3 single strands will pack together really nicely, make a nice tight braid. So collagen is fibrous. It has simple tertiary structure because it has 3 chains, and so those are related together. Technically, it's kind of between tertiary and quaternary structure, but I'm not willing to say that a fibrous protein has true quaternary structure. I'm going to call this tertiary. So the repeating sequence is glycine, something else, proline. In the case that we showed you, it's hydroxyproline. But sometimes you can have an X between the gly and the pro in the repeating pattern. But you always have a glycine, you always have a proline. The reason is when you look down the axis of it, so you rotate it and you're looking down on it now, and you make all the glycines red, look at that, all the glycines are in the middle. Every third residue must be glycine because these 3 strands have to pack together very tightly. There's not even room for a methyl group. It can't be an alanine, and also glycine helps it to adopt the phi-psi angles that it needs to. But basically there's not room for a side chain, so use the amino acid that is highly flexible, has no side chain, has a hydrogen side chain. So collagen is found in skin, it's also found in muscle, which means it is found in meat. And collagen actually makes a difference between a cut of meat and the amount you'll pay at the store for it. If you might have noticed, steak is more expensive than stew meat. And once when I was like pinching pennies, I said to myself, well, why, it looks the same to me. Why don't I take that stew meat and cook it up like a steak? You know, aren't they just trying to pull one over on me? You know, maybe I can have a life hack. But the thing is, I'm glad I didn't do that. I figured it probably was a reason why that wouldn't work. And there is a reason why it wouldn't work. The reason is collagen. Because even though they look the same, the cuts of meat, the steak has much less collagen than the stew meat. Collagen only unfolds when you get it warmer than 150 degrees Fahrenheit. If you put it in a stew and you get the whole piece of meat above 150 degrees Fahrenheit, yeah, the collagen will melt and then you'll have a tender piece of stew meat. But if you cook it like a steak, in which case you only sear the outside and the inside is rare, not above 150 degrees Fahrenheit, unless you're one of those people who likes well done. If so, you be you, but I'm going to say that most steak needs to be tender enough for me to chew, and most of my steak is better if it's below 150 degrees. Just tastes better to me. And so that means that I want a low collagen cut of meat. Not all cuts of meat have that low amount of collagen. So collagen is also found in the intestine, and so it's used for sausage casings. If you buy natural hot dogs, they are made, I hate to break this to you, but hot dogs are encased naturally in intestines. That means they are also encased in a different form of collagen. But it's all digestible to you as long as you can handle the mental image. You can handle it physiologically. The other thing, and this is something that most vegans know, gelatin itself is from animals. It's an animal byproduct, and it's a purified protein solution. It doesn't look like it. It looks like many other things that form gels, but it's a protein gel. Gelatin is when you take collagen that's been kind of partially hydrolyzed, so it's partially chopped up, and it's extracted, again, let's keep the grossness coming, it's extracted from skin, bones, and connective tissues, because that's where you find the collagen. You're not going to eat it in your steak, you're just going to try to melt it away, or you can extract it and try to use it. So food-grade gelatin is usually made from beef skin and pig hide. So if you eat pork rinds, and honestly, I think pork rinds are pretty good, but don't tell my wife, she thinks they're gross. So I eat the pork rinds in the family, and so I don't have a problem with it, but maybe you do, and if you have a problem with eating anything from animals, then you'll have to avoid the Jello dessert as well, depending on where you draw the line. It's up to you. So the thing about this matrix is you're forming a gel, and you can do some neat tricks. Like if you form the gel, if you mix air into the gel by whisking the gelatin as it's forming, you catch air bubbles in the network as it forms, and you make this light and airy Jello that they can make. So you can do all sorts of, this is biochemistry in my opinion, if you're whisking air into Jello. Here's another one that you can do. They actually took the cola and they made it into gummy. So you added gelatin to make it a gummy cola, and it's a whole bottle of cola that you do this way. And you use the bottle as a mold, and then you cut away the mold. And I think there could be a really great practical joke with this somewhere in this. But as it is, I think it's really cool, and all you need is gelatin, Knox gelatin that you buy at the store. The way this works on the molecular level, look at this if you have questions about it. But you have the hydrolyzed collagen, and you heat it up, so it's all moving around. It gets dispersed, and then it gets hydrated. So I'm going to zoom in. Here's the method, and this shows that heating is crucial, dissolving it is crucial, heating it is crucial, and then you cool it down, and it forms a network that traps the water that is around it. This is what it looks like on the molecular level. You heat it, the strands unfold and hydrate, then you cool it, the strands calm down, but now I have all this water in between. So you have protein chains, and some gelatin includes carbohydrates as well. We're just focusing on the protein here. And the intermolecular forces between the chains will hold them together. You have some kind of hydrogen bonds between the chains, and maybe some covalent. There's some covalent linkages that are still there between the chains when you do Jello. It helps it make that gel. So that's the cool thing about collagen. The other thing is I have to mention something about hydroxyproline, because you're right, you did not have to memorize it. And in fact, in collagen, some forms of collagen have hydroxylysine as well. Well, how did they get there? The one thing you know is they were not put in there on the ribosome. The ribosome only works with the 20 standard amino acids. So they must have been put in there after the ribosome by just some enzyme that hydroxylates prolines. You put that enzyme there, you get the hydroxyproline. Once it gets off the ribosome, it's called a post-translational modification. Okay, so that answers how, but why? Why would you need hydroxyproline? I've actually explained this wrong in the past. I'm not going to explain to you the wrong way, just the right way this year. It's actually a weird molecular orbital effect. So this will make Dr. Bartlett very happy, because Dr. Bartlett loves molecular orbitals, right? It turns out that hydroxylation of the proline will actually take the proline ring, which normally is in this sort of puckered endo position that's shown at the bottom of the screen here. When you hydroxylate the tip carbon, it turns the most stable form from the endo position to the exo position, which puckers the other way. This position is more likely to form the trans backbone. It's more compatible with the trans backbone. And remember how we've said that proline forms the trans backbone better as a usual way, and this really pushes it to make the trans backbone super stable. Super stable means super strong material. And so this is really kind of putting this little hydroxyl on it, flips the proline, which helps flip the omega bond, and that helps the whole thing hold together better. The angles are just better at that point. So how do you get the hydroxy? If you look at the organic chemistry of that, it's kind of a hard reaction to hydroxylate a CH2, right? So you need some help. You need a vitamin. And a vitamin that oxidizes things is vitamin C. Your body uses vitamin C to make hydroxyproline that makes your collagen strong. Therefore, if you don't have vitamin C, if you are on a pirate ship for months at a time and you run out of fresh produce, you don't get this vitamin that your body has evolved to expect. And then you start to get scurvy. Now, usually I ask if anyone knows what the symptoms of scurvy are, but I'll just tell you. You have joint aches, lethargy, and your gums start to bleed. The reason for the bleeding gums, collagen is there too. It's holding your gums together. And so you begin to fall apart, quite literally. There's some cool science with this, and I have to mention Dr. Audrey Bowden. She is actually, she's a scientist who sort of builds really cool physics machines that interact with the materials of the body. She uses polarized light to reflect off the different layers. So polarized light will reflect off the skin layer, the fat layer, and the muscle layer of skin. That means you can shine a light on someone's skin and get information about what's inside. The information you can get, what if there's a little tumor hiding in the fat layer? This will detect that. The reason it will detect that is what it's interacting with is the collagen in the skin. The collagen, if there's a tumor pushing on it, the collagen is going to be disordered, even if it's pushing too small for you to see. The polarized light will be affected by the disordered collagen, and that will show up in her technique as a signal that shows you the tumors here. Here's where it works with a mouse, normal mouse skin, nice ordered collagen, mouse skin with a basal carcinoma, which is in the skin. You see this big blue tumor that's showing up even though you can't see the tumor with the naked eye. This is just using polarized light. It's using the body as a reflector. And the reason why I mentioned Dr. Bowden here is she is a Christian who has talked about her faith and science in different forums, including this American Scientific Affiliation 2016 meeting. I'm not going to assign that, although I just thought of a way I might assign it. Okay, well, we'll see if I assign it. I'm not assigning it for test 2. Maybe I'll have it as some kind of optional thing later. Even if I don't assign it, I recommend watching it because she spends the first 15 minutes talking about her faith and growing up and becoming a scientist from her background. And then she talks about her science. Both parts of it, pretty amazing. So if we know how collagen holds together, we can maybe find we can redesign it, and we can make a material that's stronger than collagen. So one idea that scientists had for redesigning collagen is that they saw that they had the glycine here. And the glycine, again, has to pack together. It can't take up a side chain. But that scientist looked at it and realized, chemically speaking, I can put in this azoglycine thing, still doesn't have a side chain, and I think that that will actually be able to adopt the right angles, doesn't have a side chain, and the extra nitrogen will be able to hydrogen bond more. And look, that's exactly what happened. That scientist made a super strong version of collagen. When you test it, it holds together better than the regular glycine form. And when you look at the structure, it's because it's forming hydrogen bonds to these places across from itself. They aren't the best hydrogen bonds, but honestly, it helps, even so. And there's one hydrogen bond for every glycine, so it really adds up. So the important contrast to remember, keratin is cross-linked by disulfides, collagen is cross-linked by true covalent bonds. Disulfides are kind of like, yeah, they're in the covalent club, but they're just barely in it. Collagens, on the other hand, have super strong bonds, because if you look at that, for example, lysine and hydroxylysine have been post-translationally joined together with a carbon-nitrogen type strength bond. This is why the hydroxylysine is found in collagen. So collagen's just super strong, and even when you hydrolyze the collagen, those lysine-hydroxylysine covalent linkages still persist. That means the collagen is stronger even when you put it back together. It gives Jello its strength, and some scientists used it to make living bricks. Now, I believe this is the last thing that we're going to talk about, so I'm going to leave you with some text for you to read. This is living concrete that the scientists made. Concrete is sand mixed together with calcium carbonate, usually crushed shells. Calcium carbonate is made on the outside of animals because they eject calcium and combine it with carbonate. Well, bacteria also eject calcium. By the way, that's a chemical rule that is part of my book, A World from Dust, and we'll talk about that more when we get to third quarter. Needless to say, this rule is followed, eject calcium is followed by bacteria as well as by shells, mollusks. Okay? How do the bacteria make they eject the calcium, and they also make carbonate because they eat CO2 and they oxidize it. And so it means that the bacteria are eating CO2 and they're turning CO2 into concrete, or at least one of the ingredients for concrete. So scientists had the idea, let's grow up these bacteria, let's mix it with sand, and let's make concrete. And it kind of worked, but like many experiments, it didn't work as well as they hoped. The concrete was very weak. So they're thinking, what could we add to make it more strong? We need something that's biocompatible because we need the bacteria to stay alive. We also need something that works at moderate temperatures because if the bacteria get too hot, they die. So one scientist thought, why don't you go to the store, buy a bunch of packets of Knox gelatin, and mix it in? Wouldn't that be like, it would be like Jello if you mix it with water. If you mix it with concrete, won't it make it even stronger? The reasoning was exactly right. Jello is basically a protein, and it's biocompatible. So the bacteria grow just fine. And that brick that they made right there is as strong as a regular brick. But it can grow because it has living bacteria in it. So I want you to read a little more about it. Here it is right here. If I think of it, and remind me if I forget, I'll put up a link to this exact article online so that you can read it, and you can read the whole article. But just read this and think about how this can be effective not just on Earth, but effective at Mars. Wouldn't it be cool to have a living, growing concrete on Mars? You don't have to, you just mix it with sand, which is on Mars, and you have it grow in the light. You feed it CO2, maybe it takes what we exhale, and it turns it into bricks, and it keeps going from materials that are available. Therefore, you don't have to send over bricks on a rocket. You need something you can grow on Mars, and these bacteria might be able to help us do that. So anyways, that's really cool stuff. I'm going to move on to the final protein, but we will pick up here on Friday. Thanks for listening so far. I hope you enjoyed your asynchronous classes. I came in right about on time. And I will see you when we talk about silk Friday morning. Till then, let me know what you're thinking. If you have any questions, go ahead and comment or send me an email. And I will see you when I see you.